I've made my career doing dietary exposure assessment. So when Nancy sent me the email with the preliminary title here, I kind of smiled and said, this is right in my wheelhouse. I can do this one. So Philipp von Hohenheim, Swiss-German gentleman, wrote 500 years ago, probably in Latin, more than likely, something that has been translated into English, something to the effect of there is nothing that is not a poison. I'm back to where you were, Joe. We've conveniently said the dose makes the poison. Emphasize the word makes there. And really, there you go. Done. That's it for us. So, but again, I'll go back to where we just came from, sort of a little. I'll talk a little bit more of the practical, of what I think is a regulatory government world where we do have to make the decisions all the time, every day. There'll, again, be no new thoughts here. Let me see if I can figure out which button I should be pushing. Yep. The standard risk safety paradigm, I'll talk a little bit about it, and Joe certainly did. In my world, this is toxicology, this is chemistry, and the rest of it doesn't mean very much. You've got to get there to get there. And certainly I will touch on some of the same issues that we talked about, how we all need to work together to get these things done. For us, doing pre-market approval type work, which is what I've done most of my career, this is what we generally just think of as a safety assessment, and you saw it right there. Here I thought it brilliantly had bright line, gone. I did put it in quotes there at least. But I also like to say, in relation to one of the things you talked there, we get the numbers that we've now talked about, the ADI and the EDI. And as I said, you've seen it all. But I like to be a little more physical when I give my talk. So I say, this is the ADI, and the EDI is down here, and everybody's happy. There are only three cases. The EDI is down here, everybody's happy. The EDI is up here, nobody's happy. And then the real world tends to be, what do we do? And I think that's what you were getting there, and I'll also expound a little on that sort of stuff. But I am a numerical guy, so I wanted to talk about something that didn't come up. I do agree that there's always some measurable risk there because of the differences in thresholds, but in the simple world that we like to, government people like to keep it simple, 0, 0, 0, 0, 0 as the EDI approaches the ADI. You hit that number, and then all of a sudden you're in a world we've got to be concerned. You hit that line. So as a numbers guy, I've told people over the years, if your EDI, one number, if the mean of or the 90th percentile of distribution, let's be correct, is twice the ADI, how much of your 100-fold safety factor have you chewed up? Nobody knows. You could sit and pen it around for a second and say, 1%. You've taken 1% of that 100-fold safety factor away. So, and as you so beautifully put it, we need to know what those numbers are. You need to know the dose response. You need to know how all it works to get you somewhere intelligent. So I decided I should start over. I'll come in again with a slightly more appropriate talk title and continue from here. Dietary exposure modeling, which is what we do, not rocket science. It's pretty straightforward, but you do have to think about what you're doing, and Joe got into it very nicely there. We start with what we're talking about. Of course, we have to focus the problem. Are we talking about mixtures of single substances? Very typically, for my world, we're talking about single substances, but not always. In the process contaminants model, it's usually going to be single substances, although there are others. You need to know what kind of foods are being impacted. Is it across the diet or is it specific? Are the problems ongoing or sporadic? So is there a contaminant event in chickens in Belgium? You look at dioxin for one thing there. In a bigger context, you would look at it differently, but that's how you do it. How is the toxicity showing up? Is it always a chronic or is it occasionally an acute problem? There are usually only chronic problems, but occasionally we do the acute. Straightforward. You just need to know three things. The foods that are impacted, how much you're eating, and how much is in the foods. For many years when I gave a talk, I used to say, this is my science here, because I didn't have much else. As a former chemist where I used to be able to put synthetic models on there, I could always impress, but now this is all I got after 25 years in the government. But it's very straightforward. You're just looking at all the foods, food consumption, how much is in there. I put this in here for those lectures where, and this is the number of days of survey, the data comes from somewhere. And what you learn from this is the longer you've surveyed people, the lower the general numbers are. But you need to know that when you're looking at the bigger picture. And by the way, we're going to get to the break a lot earlier than we might have had I gone first. So today's talks and what we're doing about here today are the process contaminants. I have a few here, acrylamide and furan, you've already heard mentioned, PAHs, yes. Here's one you haven't seen, monochloropropane diols and their insidious esters. We've been working on that, both here and the Europeans at EFSA have been doing it. So running back through the list I had showed you, in this case for acrylamides and the polyaromatic hydrocarbons, it's foods that are cooked in dry heat. Back in 2004 when the acrylamide story was breaking, I remember having a conversation with Terry Troxel, that's a name I hope some of you know and remember, about how this was actually a good thing because it means that since cavemen were cooking food on rocks, I'm stealing your picture there, Joe, acrylamide has been in the diet of humans. This sort of thing has always been there, and that's something that is oddly reassuring. I guess by the end of this workshop and what's going on, we'll have a better sense of what it might mean. These kind of process problems, these specifically, there's a big difference between home and institutional cooking. When acrylamide has been looked at in Europe and here in the US, the food industry stepped in to do what it could, changed how you produced your potato products, for example, a little less crispy, a little less brown. You see reductions in the acrylamide levels in those foods. But when you actually send those packages home and people cook them according to the package and they take it out of the oven, Mom puts it, yesterday being Mother's Day or the day before, puts it out on the table. Everybody wants them brown and crispy, and back into the oven they go, and up goes the acrylamide. EFSA has just finished redoing its reevaluation of acrylamide, and this was one of the things we talked about a lot. It's a huge portion of the uncertainty of that analysis. Concentrations of these materials in food are highly variable but predictable for these. In the case of furans and the MCPDs, it's a little more random. You're not quite sure what you're getting there, and these are not as well-developed stories as these two are up here, especially from my perspective. Again, these are ongoing events, so it's always been there. It'll always be there. It's in all foods potentially. The model that we use at FDA for acrylamide now has 68 different food categories with acrylamide numbers in it, so it's everywhere. Although you can definitely look and see specific subproducts. I used to say euphemistically that potatoes were the killer when we were talking about acrylamide because chips and fries always had the highest levels, and combined with the amount of the food you were eating, you were always getting the highest numbers there. Talk a little bit about that later. These are, of course, all chronic toxins. I'll start here with the general, and I'll come back to a little more specific, and you will see a couple of these slides more than once as I come back to them. As was mentioned, you really do have to have a good amount of data to get a good answer, and you have to know what the uncertainty is. So again, this is not a new thought, but it's pretty easy when you look at some of these things to discern whether or not we are guessing or not. And in some cases, it is an educated guess, but hopefully we can go down the road and do the kinds of thing that this work group is going to bring us and get to a better place. When I put this bullet up the first time, I had a brilliant idea that has somewhat slipped from my mind, but Joe, you kind of brought it back to me, and I think it goes to the interaction between looking at the distributions of exposures that you can develop with the new approaches, the MOE type approaches, and benchmark those approaches. I think this is exactly the way to go. I completely agree. See, here I thought I had that. I was going to come out with a point, not a point estimate, dashed, but I agree, there it is, and I've been saying this for years. The answer to the question is never the exposure is. You always have to talk about what you were talking about, where you are, what it means, the shape of the curves. I'll show you some pictures later that will beat my point to death, but you do need to know these sort of things. And to go from the other end now, where we're talking about mitigation, this is where the managers and the assessors need to have conversation. I know the paradigm originally called for these separate. We do our risk assessment and the managers do their management separately, but you just can't do that in the modern world. There's too many factors that have already been mentioned. I'll give a couple of examples here where if you want to reduce exposure by putting limits on periods, well, then you have the economic impact because you'll be losing foods. If you're just going to do reductions as has been proposed, for example, for acrylamide, well, then you have to see whether or not you're actually getting any benefits. And again, this is something we've already talked about this morning. So I'll talk about some old friends here. I've been doing these for a long, long time. Acrylamide was popped up in 2002, as I'm sure you all know. It was already known as a carcinogen. At that time, the mechanism was unknown, but we've gone a long way to know what's going on there. And of course, it was also not known what was going on. In fact, I think when the Swedish group first released their results, there were only about 8 food groups that they were mentioned. As I've mentioned, we've got 68 different categories now in our analysis. Furan was to me the fair-haired stepchild of acrylamide. It came out about a year and a half later at FDA, and it was in the shadow of acrylamide, but our analytical chemists were detecting it in a lot of the things we were looking at, so they finally figured out what was going on. At that point, it was well known that it was a carcinogen. Again, no known mechanism at that time, and it's not all that much better, although clearly you're rearranging carbohydrates. And the extent of presence in food was not known. The ADIs were out there. We had already done that bright line work, mostly because there were indirect additives that contained the material and it was a contaminant, specifically for acrylamide. EDIs were whipped up quickly, and if you want to talk about doing science based on little data, if you go back and look at that first Swedish analysis, it was really tiny, but it got it out there. Later that year in 2002 or 2003, I'm not exactly sure now, I don't remember, WHO put together an expert work group which had a little bit more data. And FDA was a little slow on this. It took us about 2 years to gather enough data before we felt comfortable enough to publish our first exposure. But the interesting things were that everybody was getting about the same number. And it was all in this 0.5 to 1.0 range. In 2000, can't remember which year it was now, 5 or 6, JECFA took on the project, and we came a little more conservatively to 1 at the mean microgram per kilogram body weight, up to 4 at the high end. And 5 years later, when we reevaluated it, we decided that those numbers would still stick. Our Furan assessment was first done in 2004. We went back as we got some more data. Depending on the crowd, I tell a little story here. I'm a big believer in appropriate numbers of significant figures. And I tell my young people, some of whom are here in the room with me, if you see an assessment that's got 2 or more significant figures, start asking what's going on here. So when I was doing these Furan things, as I was getting more and more data, I would just go back and stick them in the model. You know, once you've got your Excel spreadsheet set up, it's nothing to it. So I was adding more, and I had Excel set up, so I only saw 1 significant figure. So it was 0.2, 0.2, 0.2. One day I got, and this was from Nega's group, I'll tip forward to the future speaker. I got a small amount of new information on a fairly minor food, and I plugged it in, and up came 0.3. And when you tell somebody that the exposure went from 0.2 to 0.3, which is a 50% increase, you know, a little crazy. So, you know, you drag your column over, and what happened was it had gone from 0.2499 to 0.2501, right? So you have to be a little careful when you do these things in the real world. There are considerations to what's going on out there. At that point, I acknowledged that we could say 0.25 and leave it at that. So I wasn't happy about the second one, but we did it. What I'm referring to here is exactly what Joe was just talking about, how we do need, this wasn't enough. That was the bottom line, and I agree, absolutely. You just can't take these numbers and say the bright lines are enough, so you have to do it differently. We prepared all of these distributional models so that you can plug in numbers and take a look at the mitigation impacts, which I will certainly get to a little bit later. And the benchmark dosing things is what I was talking about with these new tox studies, and taking classic methods but using the data in new ways for getting a better look at risk. So I don't think we've heard a definition of mitigation measures necessarily, but there you go. Reduce risk to a substance in food. And it is a complex question. Who gains, who loses, who pays, who talks about who has to pay? There's lots of questions there. This is not the assessor's problem, of course, and I agree, it goes up the road. And you need to be able to talk to the public. You see it every day out there now. I mean, we don't go through a week where we don't hear a story from a blogger who has an opinion, and out it goes. And if you're not in control of the science, the discussion of the science of the story, you can lose it. I mentioned I would bring slides back. Here's the first one that comes back. To this, I've added, for the purpose of this group, that we're going to talk about managing exposure to reduce risk, if you can manage exposure to reduce risk, and Joe touched on that. And as I mentioned earlier, it depends on what we're talking about. Typically, for the kinds of process contaminants we're talking about, it wouldn't be upper limits in economic impact, but it would more be the cost of what we're doing. So here's the first generic picture I will show. This is a classic dietary exposure distribution. I could name pretty much anything. It happens that this actual slide was from my acrylamide analysis from 2006 or 7, I think, actually. So these are the actual numbers, 1 and 2 micrograms per kilogram body weight. Dietary exposures for commonly consumed foods are almost all log normal-ish. Counterintuitively to most people, the mean is typically about the 70th percentile. It's not in the middle, as you might guess. It's certainly to the right of the mode. The 90th percentile for almost everything is almost twice, more or less, 2.1, 1.9 times the mean. So these are nice rules of thumb you can use for pretty much anything. Things that fall out. Foods like shellfish, where you just don't consume them in the normal way. Those things don't come in here, but bread, milk, meat, vegetables, all look sort of like this. Interestingly enough, it's also true for other things. So this, you'll notice, is the same picture, but with a different title. So when we've looked at not a process contaminant, but a natural contaminant, aflatoxin, now instead of what you saw before, which had number of people and how much they're consuming, now you see number of peanut batches and how much aflatoxin is in them. Looks kind of the same. Or you could look at methyl mercury in fish, another thing, looks pretty much the same. I've beaten you to death with this. It's the same picture. It makes it easy from my perspective to at least qualitatively show what's going on. When you talk about mitigation, are we talking about here or here? And that's to where quantification comes out. So here are some real data. I had to do something that had something to do with current. We have been looking this past few months at republishing our acrylamide exposure estimate. What I have here are the top 20 foods, and the individual numbers aren't so important. But the point I want to make here is if you look, even the top contributors are only giving a small amount. So when you have 20 foods that end up giving you 90%, you can quickly realize that changing one or a few of these may not have much of an impact. And I had originally made this slide almost 10 years ago now. It's, I've updated some of the numbers. If you look at the mean, and please again forgive the second significant figure, if the mean is 0.44 with a 90th here, if you could wave the magic wand and completely remove acrylamide from French fries, let's say, your mean drops down to 0.37. There's two ways to look at this number. I always talked about it. If you're an optimist, you say, well, we lowered it by 0.07 divided by 44. That's a 15% reduction. If you're me, you say we went from 0.4 to 0.4 and you can't really measure the difference. So if you can't measure a difference, and I think Joe touched on this a little bit, you have to be able to decide what you're going to do with it. And as you go down the list, so second, third, fourth, fifth, you know, you're going to get a smaller and smaller impact from any given food. So you might just say, well, if we could reduce it 20% in all foods, we would reduce it by 20%, and that's correct. But let me see, is it here? No, not yet. You still have to move the mountain, which is what I'm saying here. What we're concerned about, what we need to be concerned about as regulators is what is the impact of these things. It's not enough to just say something is bad, less of something bad is good, so we'll lower it. It's got to have some kind of impact. There's just too many considerations that need to be brought to play. Small changes don't make much of a difference. I want to take a look. I'll show you last slide here, and I used aflatoxin because it was the best example. About 10 years ago, we did this for JECFA, and the question came in, if we reduce the upper level from 20 to 2, would that make a significant impact? So again, as I mentioned earlier, when you're looking at 20 out here, the area under this curve here is the total amount of aflatoxin you're taking out of the diet. This turns out to be 0.05% of the total. So by putting a limit of 20 on there, you're not actually accomplishing very much. You might as well just let all the peanuts go. So to go down to the 2, which was in here, now at least you've got some color under there, but you're still only looking at 1 or 2% of the overall amount. So now you've made a significant reduction in this number, but not a big number here. So where do you need to go to finally start moving the numbers? Well, you need to get down to about 1 part per million, which is closer to your mean, and you remember I was explaining the mean 90th. Now you're losing 40% of your peanut on a bad year, not necessarily a clean year, but a year that's got a lot of aflatoxin and peanuts. That's a lot of number, and that's a lot of peanuts that somebody is going to have to take care of. So I guess that's what I just said here. I'm sorry. What you see there is also true for methyl mercury in fish. So what are my take-homes that are even slightly different from the ones that we've already heard, and necessarily there aren't any. But I'd like to say that you can do dietary exposure modeling, and it can be useful and will be helpful in this process. The uncertainties, you got right up there to the end and you hadn't talked about something else, and then you talked about uncertainty. So, all right, true, and I do believe that we have to be careful about both describing and quantifying uncertainty. Dietary exposure uncertainty is difficult. When you talk to an animal toxicologist, benchmark dose type kind of stuff, you got 50 animals, you look at your numbers, you get error bars. Dietary exposure, I'm asking, not me, but let's say NHANES is asking people to write down what they eat. We know there are errors there, but it's not that kind of statistical quantification of error. I was at an ILSI Europe meeting about 5 years ago, and Wout Slob, European modeler, was there saying, well, can't we put some bars on there? Can't we just put a factor of 3 around it? And that's meaningless when you're looking at this stuff. If I tell you that the average consumer of potato chips eats 20 grams a day, I'm not going to say the bounding limits are 7 and 60, because that doesn't really mean anything. It's not like you're putting it in the middle. So it's a little tricky to do this sort of thing when you're looking at the dietary exposure parts. But I've also said, as a chemist in a world of toxicologists, here's where I get to be a little arrogant, I can guess what the dietary exposure is, and I think I'll do a better job than using animals to tell me what's going to happen to a human over a 70-year lifetime. Anyway. So the bottom line that you get to here is ultimately the risk managers are responsible, and I'm happy to throw my hands up and say I'm not responsible there, but it's not easy, and it's going to be a difficult process, and I hope that this work group helps us get down the road. And now I will say thank you. And I guess we'll wait till later for the questions.